All right. So, as I mentioned before in class today, we wanted to um, kind of get through a few more of these slides so that we can finish this at our next meeting for class. And then we can move on to the next segment. So, I just wanted to start off with this slide again, just to remind you, we have DNA and R, and we have DNA here. We have two strands, the 5' 3' strand that is considered our coding strand. So as we go through here, if this is an A and a T and a C and a G, then like our RNA, right? The RNA that's coming out should also be A, U, C, G. And so in order to kind of think of this, right, we therefore need to consider that the opposite strand of this, if our DNA is A, T, C, G, our complement strand would be T, A, G, C. Now we know that RNA polymerase has to make the enzymatic cleft and the active site of RNA polymerase makes this particular duplex system because it's reading the template strand, right, in the 3 to 5 direction, and it's priming, it's polymerizing in the 5 to 3 directions. So you have the 5' end, right, and this will eventually become the 3' end. And so if with this particular example, if this is A, T, C, and G, then these light blue sequences would have to therefore be T, A, G, C. And because the complement strand is the one that's being read, it allows the RNA, because it's making this hybridized duplex, to then have each free nucleotide being added is then the complement to the to the template strand. So therefore making it A, U, C, and G. And that's what we get, and that's what we want. So RNA polymerase binding, that is going to be our initiation of transcription. The next step of transcription is elongation. Elongation is that process by which RNA polymerase reads that template strand and begins to elongate the mRNA that is being created. And again, we have different species of RNA, right? We have tRNAs, and they are involved in translation, but they also look like T's, so therefore they get a name. And the RNA that comes off of these, if they code for protein, so protein coding, these are considered to be messenger or mRNAs. And then, of course, there are different kinds of RNAs like siRNAs, CRISPR RNAs, non-coding regulatory RNAs, and so on and so forth. And so we make the RNA as it comes off the DNA template strand. And again, downstream of RNA polymerase, you're going to have things like topoisomerase and helicase. And as the RNA comes off of the DNA and you get religation of the DNA back to its normal form, you have polymerization, religation. So you have ligases and gyrases, right? Gyrase that help rewind that DNA. So as you're making the RNA and it's coming off the DNA, at some point there has to be a terminal stop. There has to be an endpoint. And termination of transcription in both in eukaryotes and prokaryotes highly depend on another important structural region of an RNA, and that's the 3' untranslated region. So the 3' untranslated region is a region of the DNA that has sequences or sequence structure by which the RNA polymerase and the transcription complex is compromised in its ability to bind and polymerize more mRNA. And this is highly dependent on whether or not it is a row factor independent method of termination or row dependent method of termination. So what is row? Row is a translation factor. It's a small protein that basically guides along the mRNA and moves along in search of a row-dependent transcription sequence. And the row-dependent terminator, or the termination sequence, typically has a secondary structure, which is made by a bilateral palindromic sequence separated by a small spacer, and that allows for the creation of something like a hairpin. And that hairpin, when it is reached by row, actually makes contact with RNA polymerase, and it does so because the secondary structure, because remember, right, we're linearizing, right? We're going from our helical structure like so to a simple ladder structure. So if we have any secondary structure at all in any of the sequences, this causes the RNA polymerase to pause. And that pause, or the slowdown of RNA polymerase, allows for row to catch up. That's kind of the function of that row-dependent terminator. It causes RNA polymerase to slow down, and when it slows down, RNA polymerase then is engaged by row, and row causes a conformational change to RNA polymerase, causing it to vacate off of the DNA and therefore terminating transcription. Row independent termination is simply caused by a polypyrimidine track. So polypyrimidine tracks on the RNA caused by a polypurine track in the DNA causes instability of the ability for RNA polymerase to maintain engagement with the DNA. So the ability to get RNA polymerase through a polypurine track to create a polypyrimidine track is by and large really difficult. So what ends up happening is when the RNA polymerase reaches this polypurine track, creating a polypyrimidine RNA, it can no longer maintain that site. And as I mentioned before, there's no poly A tails in prokaryotes. So what ends up happening is when it reaches this, it actually terminates transcription in the beginning stages of that. So what you're left with is only a very small segment of that polypyrimidine track in that coding sequence. In the non-coding region of the mRNA, and then you get because again, they cannot maintain engagement with that DNA, so it can no longer engage with the RNA. So that whole complex segregates and disassembles, dissociates. That's the word I'm looking for. Sorry. And then you get RNA transcription termination. So this is what that stem loop looks like for row independent termination. All right. So again, in both cases, you have these secondary structures that are made by the RNA. The row independent terminator again forms a secondary structure by way of a palindrome, right? So GG, CC, U, A, G, C, so on and so forth. And then there is a small spacer sequence, which gives rise to this hairpin, just like we saw with the CRISPR locus. And those inverted repeats or the inverted palindromes are really critical for the ability to make these stem loops. And again, the stem loop in the RNA runs upstream of the run of the polypyrimidines, and therefore in the DNA, it runs upstream of the polypurine track. And that causes a severe pausing and then eventually instability and dissociation of the transcription complex. So once we get the mRNA, we've seen this slide before, so I won't belabor it too much. Right, when we have the prokaryotic mRNA, right, we have the 5' untranslated region. This is not including the promoter. The sequence for the promoter, like the TATA box and the Pribnow box, would be somewhere over here if this was the DNA. So this is a different amount of sequence between the 5' untranslated region. And every 5' untranslated region has this really important sequence known as the Shine-Dalgarno sequence. So whenever you're thinking about cloning bacterial genes or bacterial operons into a plasmid, for example, one of the really important concepts for that transcription and translation process when considering how to make said cloning plasmid, we always have to think about where the Shine-Dalgarno sequence is and make sure that we're including it. So that way we know that we're getting critically translation. So the Shine-Dalgarno sequence is a consequence on the sequence by transcription. So therefore, it's used during translation. And we'll talk about what that is in a few minutes. And again, most prokaryotic mRNA is multicistronic, meaning it has multiple genes, which encode for multiple proteins, and they each have their own individual start and stop sequence with small interloping sequences in between. But the purpose here is that all three of these genes are controlled by a single promoter and typically a single set of regulatory regions within the promoter. So therefore, they are what we call an operon. Eukaryotic mRNA, again, has much larger 5' and 3' untranslated regions. The 5' and 3' untranslated regions have these very elegant and very elaborate secondary structures, which are really important for the translation and mRNA stability purposes. There is the 5-methyl-7 guanine cap, which is really important for triggering nuclear escape of the mRNA and migration to the ribosome. And then there is the ever present poly-A tail, which is unique to eukaryotes. Now, as I will remind you, all eukaryotic mRNA is monocistronic, so we only encode for one gene per mRNA. So even though you might have an mRNA of similar size, the mRNA still only encodes for one protein rather than 3, 5, maybe even 7 genes. Now, what are some of these other things that you see here? There are things called IRIS sites and IRE sites. These are called internal ribosome entry sites. And so what these are really important for situating eukaryotic mRNA into the ribosome. So the ribosome, the different ribosome catalytic sites, the initial binding site binds around these sequences, and this sits in an internal pocket in the ribosome in order to allow for the mRNA to sit correctly in the correct orientation to allow for the ribosome to find the start methionine and then proceed with translation. So as we'll remind you also that the ribosomes between eukaryotes and prokaryotes are very slightly different. Prokaryotes produce a 70S ribosome, whereas eukaryotes produce an 80S ribosome. This is because the large and the small subunits are distinctly different. in the amount of rRNAs and types of RNAs, as well as polypeptides that create them. So these differences in combinations result in slightly different chemical and structural profiles. But I only say slightly different because, again, the whole purpose of a ribosome complex is for the catalysis of aminopeptidase and aminotransferase reactions in order to generate a polypeptide chain. So if the structure was radically different between the two, then the chemistry probably wouldn't be the same, or the ability to create the polypeptide would be lost. And that is really important to understand. So once the mRNA is created, again, we have this process of translation, and again, transcription and translation are extremely how would you say, coupled, excuse me. They're extremely coupled between within prokaryotes and not so much with eukaryotes. And this all has to do with the fact of the lack of nucleus and the accessibility of promoters inside of prokaryotes. So in every mRNA, the Shine-Dalgarno sequence, as I mentioned before, it's a part of every mRNA. The mRNA that has the Shine-Dalgarno sequence is really critical because the 16S rRNA that exists in the small subunit of the ribosome, seen here, the Shine-Dalgarno sequence and the 16S rRNA here, are an exact complementation match, meaning that there's going to be an initiation factor which binds to the ribosome. That's going to cause a conformational change, which will pull the 16S rRNA up to the binding groove for the mRNA. And then the ribosome binding site, the Shine-Dalgarno sequence, will complementarily bind to that. And then what you get is the distance between this initial A and this U is a divisible by 3 number. So meaning that that when the mRNA binds, you can actually have the ribosome fit codons from the Shine-Dalgarno sequence on through until it reaches that first start methionine. And this whole idea of structure and spacing and distance is why when we talk about indel mutations or SNP mutations or missense mutations that exist in the non-coding region are really critical, because if they occur in the promoter, if they occur in a regulatory region, if they occur in the Shine-Dalgarno sequence, it can significantly impact the ability to express that gene. Because again, the whole concept of gene expression is protein expression, protein function. So, and that's all housed from that concept, from central dogma, DNA to RNA to protein, right? So from here, when we get this feature where the Shine-Dalgarno sequence sits down on the 16S rRNA, the secondary initiation features the secondary initiation factors are bound to the ribosome. This is the triggering of translational initiation. So translational initiation begins when this occurs, and when all of these things are assembled. This is actually what triggers the large subunit. So this is the large subunit to actually sit down on top of that binding groove and allow for the beginning or the assembly of the different catalytic sites or recognition sites within the ribosome. Now, within the ribosome, there are 3 really important centers. There are, it depends on how you mnemonically want to remember them. If you read them left to right, they spell, the Environmental Protection Agency, the EPA. If you want to read them backwards, and that's the order by which mRNA is read into the ribosome, it reads APE or APE. I always think of ape because it helps me kind of reminds me of the directionality of the ribosome. So in each of so once the Shine-Dalgarno sequence has been read, and um and then you get to the first AUG, what ends up happening is the AUG is first decoded, because this would, so over here would be the Shine-Dalgarno sequence. And so the mRNA is moving in this direction, so to speak, and the ribosome is moving in this direction. So the AUG would be read at the A site. This is also called the amino ACL site, or the decoding center. So it decodes what anticodon, right, with the tRNA. So each tRNA is ester bonded to an amino acid, and therefore each anticodon is very specific to a codon, and then that codon gives us the codex, or the decoding of what amino acid should be brought to the amino acyl site. Once the amino acyl site is bound by a tRNA, this causes the large subunit to move. And when the large subunit moves, it then shifts this codon and the tRNA to the P site, or the peptidyl site. And this is where you'll get a peptidyl transferase reaction, where you'll create a new peptide bond. And then once this occurs, the next tRNA will bind to the next codon. And then when this occurs, again, the small subunit will shift, and then this will cause the large subunit to shift. And so this first tRNA then moves to the P site, then moves to the E site, or the exit site, where there is not enough ribosome cleft and interface for the anticodon to remain on the mRNA, and the amino acid has been successfully transferred to the growing peptide chain, or the growing polypeptide chain. So the tRNA then very simply just falls off or dissociates from the complex. So the energy that is used, and the ability to transfer these amino peptidase, sorry, the peptidyl transferase reactions, is through not through ATP, but through GTP hydrolysis. So here it is up here, GTP hydrolysis. And this is what triggers both the reaction to allow for the function of the peptidyl transferase function, as well as the ability for the ribosome to continuously migrate along the mRNA. So, in each time that amino that peptidyl transferase is causing the basically the reformation of an acyl bond between the different amino acids. And this is what then allows for the peptide to remain intact as it grows and grows and grows and grows. So over here, you'll get kind of this kind of sense of how this works. So A site, P site, E site, and removal or dissociation of the tRNA as the ribosome moves along. But I like this cartoon because it shows you that all along it's the ribosome, right, that's moving along the mRNA. The mRNA really doesn't move per se, except as a consequence of the movement of the ribosome. Now, each codon, which responds to a different anticodon, will respond to the generation of a different amino acid based on the codon that it presents with. And as I mentioned before, there are 20 canonical amino acids, and there are, I haven't done the math on this, but you basically have any combination of U, C, A, and G in three letter combinations, which is a great deal more than 20. So what this results in is having multiple codons. So for example, these all CUU, CUC, CUA, CUG all result in generation of leucine, but so does UUA and UUG. Now, there are 3 distinct stop codons, which are only produced by a single amino acid, that's right, by a single nucleotide codon. And they are UAA, UAG, and UGA. And as you might look at this and say, well, wait a minute, we talked about this previously, about transitions and transversions. Well, here's serine, UCA. It doesn't take much to go from UCA to UGA, for example. Or, you can have UAU or UAC for tyrosine, not so far away from UAA. So important to also remember that proteins are always created. The very first amino acid that start methionine defines its N terminus, and the end of that sequence is the C terminus. And this has to do with which end of the amino acid is facing that side of the chain. So N referring to the amino terminal end, and the carboxyl end being the carboxylic acid that's on the other side of the amino acid, the amino acid base molecule. So as translation proceeds and the polypeptide grows, this is translational elongation. And here it is in all its glory, where you have the growing peptide. And finally, you're going to get to those stop codons at the end of that transcription, because again, transcription will end, past the first stop codon, because, you need to have the 3' UTR and such. So once this occurs, and the stop codon reaches the amino acyl site, this will bind a very specific set of factors known as release factors. And release factors then actually trigger the migration of GTP and GDP. And so GTP and GDP come together. GTP is hydrolyzed, and the hydrolysis of GTP allows for the release of the polypeptide chain, and you use GTP a second time to release the release factors off of the ribosome. This allows for the polypeptide to be released to then begin. So this leads to the nascent peptide. That is the primary structure. It goes through the various structure foldings that we've already talked about before. And then prokaryotes actually undergo a really cool kind of process where they will actually use elongation factor and other release factors in a complex with the combination of GTP energy to dissociate the entirety of the large and small subunit complexes. And this allows for reinitiation of the small subunit with other nucleic acid species, and eventually the removal of the terminal tRNA. So as I mentioned before, there is a great number of different levels of control. And we talked about this in class today about there being control at the level of transcription. This is where 90 to 95% of all regulation occurs, because again, this is the most upstream function of gene expression. So that is by far the easiest way to control by energy conservation standards expression of genes. There's also translational control, which is, again, things like codon bias, rate of translation, the structure of the 5' and 3' UTRs, and then post-translational control, which is the control of protein activity, which means that you are making proteins at the level of translation, they come off and they fold into their globular structure as an inactive or apoenzyme or apoprotein, and then they wait for that stimulus or that coenzyme or metallic cofactor to activate. Now, it's really important to think about this from a comparison standpoint, right? In eukarya, there are a ton of different levels of control, including chromatin remodeling. This is the histone binding, right? Or the ability to open the DNA in order to get access, or the accessibility of the promoter. Then there's the structure and function and migration of the mRNA, including splicing and intron removal, and then migration to the ribosomes, and then translation and post-translational modifications, which are all but these three here, transcription, right? So transcription, translation, and post-translational modifications, as well as things like epistasis, epigenetics, non-Mendelian modes of gene regulation, are all mechanisms by which genes are regulated both in eukarya and prokarya. So when you think about this, a lot of the differences that we talked about between eukaryotes and prokaryotes, a lot of the micro differences, like having chromatin, having histones, having a nucleus, having an endomembrane system, all result in these subtle differences in the systems of transcription and translation. So when we talk about genes, the genes and gene expression in the level of protein basically break down to two distinct modes of regulation. There are those that are constitutively expressed, meaning that they are always transcribed at the same level. So they are actively transcribed under all experimental conditions, meaning that they don't change. So these are typically like housekeeping genes, essential genes, genes that the cell might spend a lot of energy to produce, but are really essential for cell survival. So to thread that middle point is to continuously express them, but at a level by which they don't have to stress the cell to produce more of. So if you have genes that are not being constitutively expressed, if they're not being expressed at a constant rate, that means that they are being regulated. So regulatable genes, so these are both regulatable genes that are regulatable or can be controlled in their expression, are either induced when they are transcripted and translated at a higher level in response to some type of factor, and repressible. So if you can go up in expression, you should be able to go down in expression in response to a specific signal. In most eukaryotes and prokaryotes, as we talked about before, there are typically, as we see here, there are promoter sequences, and then there are upstream sequences, which typically result in the formation or having cis or DNA factors, DNA sequences that are control elements. Call them control elements. Now, many of them exist upstream of promoters. Some of them can exist downstream of promoters, such as operators. And they combine to both enhance or suppress transcription. Again, by binding the DNA, just like anything else that binds anything else, proteins that bind DNA in a small environment are going to change the ability for other proteins to bind essential parts of a DNA sequence. So some of the control is steric hindrance, so just simply being in the way. Some of it is structural, And all of it, in some way, shape, or form, has to do with both promoter availability. So promoter availability, as well as the ability for RNA polymerase to bind, like the availability of the promoter, but also to read through. So any of these things that can affect promoter availability or the ability for RNA polymerase to do its job can actually regulate gene expression. Now, in a lot of operons, in a lot of operons in most bacteria, they have a very similar setup where they have 5' and they have 5' regulatory sequences that occur here. The promoter, which is where RNA polymerase is going to bind, and then they have these sequences called operators, which is where they get the term operon for. And operators are typically bound by some form of regulatory gene that exists usually within the same locus, within the same gene space in the DNA. And so the idea is that as these are being transcribed, independent of transcription of the genes that they are regulating, this creates a regulatory protein that can either bind to these regulatory elements or to the operator. And again, the regulatory genes, we call them, in total, the class of them, we call them transcription factors, because they are factors which regulate transcription, or more accurately, some people call them transcription regulators. And the actual regulator, the actual protein that does that job, is the protein that actually binds the DNA and causes that regulation of transcription. There can be a number of cascades that we talked about. There are a number of signals which trigger regulation of genes, right? So again, go back to this, right? Repressible and inducible genes, they're being induced or repressed based on the situation, based on the environment. Some type of signal or factor is causing that level of regulation or induction or repression. So that means that there's going to be a signal, right? There's going to be a signal, so there's going to be a signal that eventually triggers somewhere downstream of this system a regulator. The regulator is what binds the DNA and then influences either repression or induction. And of course, like most operons and most mRNAs, they are multicistronic, which means that you get a singular mRNA, but you get multiple proteins that are encoded for by that mRNA. So one of the very common systems by which these genes are regulated are through feedback loops. So the activation or inhibition of most regulatory proteins have to do with different kinds of feedback loops. So those signals from what the cell needs versus what the cell has access to, environment versus intra and intracellular signaling. And so there are two types. There are positive feedback loops and negative feedback loops. And so in the positive feedback loop, you have an input into the system. The system generates an output. That output then feeds back to the system, which generates more output. So you're getting an increase in output in this case. In negative feedback loop, the input goes into the system generates an output, but then the output feeds back to the system and, like the name suggests, negatively or downshifts the system's ability to produce this particular output. So one of the very well studied and characterized pathways of this is the biosynthetic pathway of tryptophan. We talked about this very briefly when we talked about regulation as it relates to physiology. So again, we're just going to go a little bit more in depth here. So tryptophan is a biosynthetically created amino acid. It's an aromatic amino acid. And so it requires a number of different enzymes to go from its generalized precursor to generate a hexose and pentose ring as its R group in relation to being then acyclically bonded to the amino acid core molecule. And so tryptophan itself makes for a regular negative feedback loop. Now, not for nothing, but in nature, negative feedback loops exist far more frequently, like 9 out of every 10 feedback loops, if not even more than that, maybe 99 out of 100 are negative feedback loops. The existence of positive feedback loops as a particular loop is actually very low. So when we talk about induction of genes can be induced by positive feedback, but it's very rare. Usually it's from an outside system that is important for induction. But repression very much feeds into or is from feedback loops. And this is why. So remember, cells have a finite amount of energy, and they can always assimilate more by breaking down more glucose, but glucose isn't always available. So being able to conserve energy in a way that makes sense is really important. So this is one of those cases where tryptophan is an essential amino acid. It's found in a lot of proteins, right? But again, if a cell needs this much tryptophan, and so this is what it needs, and it then makes this much tryptophan, so it has what it needs, well, then you have a situation where you then might get, you might make this much tryptophan. So this tryptophan, that's more than what the cell requires, ends up going back into the cell and then binding to both the transcription regulator of tryptophan. So here's the transcription regulator, which would bind to the operon of the tryp gene to allow for transcription. And by binding tryptophan to that regulator, this causes the regulator to prevent transcription. And so you get downshifting, you get loss of transcription of the enzymes, right? The enzymes required to biosynthesize the different intermediates of tryptophan. Now, the interesting thing is that the target here really is all of the genes, but the hallmark of this is to not spend any more energy than we need to. So just like transcription is really important for gene regulation because of energy conservation, so is targeting the very first enzyme of a biosynthetic pathway, because then you're not spending all of this energy to get to these other intermediates and then stopping the biosynthetic pathway when you're almost there anyway. So the feedback inhibition works by, again, taking this excess tryptophan, and it then binds to enzyme 1. And again, I will remind you, these are typically, so like here's the enzymatic cleft, right? They might bind to like this kind of cleft here, and the allosteric site. So most of this is allostery. And when you have allosteric binding, it causes a conformational change to the protein, therefore occluding the active site and preventing further catalysis of that biosynthetic pathway. So you prevent making more tryptophan than what is necessary, and you prevent making more enzymes that would spend more energy that isn't required. So induction and repression can actually be caused by feedback loops, as well as molecules related to the breakdown or utilization of certain molecules. And so the two different types, so we talked a little bit about repression being in the negative feedback way. the binding of molecules in a positive feedback loop or in an inducible way happen to come in two different forms. There is positive inducible and negative inducible ways of inducing transcription. And this all has to do with where the particular thing is binding. So in the case of positive inducible activation of gene transcription, when you have an activator protein, so in the absence of the activator or the regulatory factor, which enhances transcription from binding the regulatory elements in the DNA, you get no transcription. But then when you add the inducer, when you add that molecule to the system, it causes a conformational change to the transcription factor. It binds the DNA element, allowing for transcription. Negative inducible is actually when you have a repressor. So the repressor here is bound to the DNA. It is not allowing for transcription because it is probably stereo. So RNA polymerase probably binds up here. So it's preventing RNA polymerase to go forward. And so in this case, because it's negative induction, you're inducing by negating the repressor, and therefore induce, because if you're negating a repressor, you're inducing transcription. So by adding an inducer, it causes another conformational change to the repressor, pulling it off of the DNA, allowing for transcription. So this is just another example of different ways that we regulate genes. And again, there are two different subtypes. So, if genes can be induced or repressed, there have to be, therefore, especially in prokaryotes where we don't have histones and nucleosomes for a way of regulating transcription. Transcription is almost entirely regulated by signals that induce expression and function of transcription factors or transcription regulators. So again, binding transcription factors, again, if you have an activator, just as an aside, if this is, say, from a phospholipid system or another set of systems, you don't need to have necessarily a molecule of induction. You could have a signal of induction or repression. And in these cases, you have a signal from up on high. There's a signal transduction that occurs. This allows activators or repressors to bind sites in the DNA. And by binding sites in the DNA, they either inhibit RNA polymerase from reading through, or they basically help the assembly of the transcription complex and RNA polymerase onto the promoter, i.e. making the promoter more accessible. So again, this is all about promoter accessibility and structure accessibility of the promoter for RNA polymerase. And for what it's worth, activators also are important for not only engaging or enhancing or synergizing the creation of this complex, but it also attracts more RNA polymerase and other factors of the transcription complex to the side of the promoter. So you get this like constant barrage of RNA polymerase that's binding to that promoter because of the presence of the activator. Whereas when RNA polymerase is stalled or is prevented from getting past a certain site to get to then the promoter or the start AUG, the RNA polymerase complex cannot maintain its normal structural integrity, and so the complex dissociates, and therefore so not only does it halt RNA polymerase, but it also then disassembles that complex and then blocks RNA polymerase from doing its job, and therefore in halting or at least lowering the ability to perform transcription. Need a quick moment. Sorry. Okay. So, when we're thinking about this, some examples of this, is you know, so we've talked about, so far we've talked about this concept of an inducer, or a chemical, a chemical modifier which causes induction or repression. So we talked about uh, inducibility by inducer addition. The same thing can occur with chemical, so not necessarily cellular signaling, but chemical composition, or chemical environmental stimuli, such as like tryptophan. So here's an example of that pathway I showed you. So this pathway, right? So the left side, sorry, of this pathway, this is the regulation of gene expression. Tryptophan can bind a repressor. That repressor binds to the operator. This functionally prevents RNA polymerase from reading through, and that stalling and movement of RNA polymerase up against the repressor in the operator sequence, where it can't move, causes the polymerase complex to dissociate, and then completely halts transcription, or at the very least suppresses transcription lower than its current state. On the same time, there are negative induction pathways, and the lactose operon is a really cool kind of introduction of how you can have both positive inducible and negative inducible events happening at the same time. And these can affect transcription at various rates. And this kind of gets to this concept that there are things like operators and upstream regulatory sites, and both of them, both the upstream regulatory sites and the operators, can be influenced by chemical and environmental stimuli, or intracellular chemical compositional stimuli, as well as transcription factors and regulators that come from types of phosphokinase relay systems, like we saw with quorum sensing, can affect things binding to this. So basically, what I'm saying is that transcription in most bacteria is very, very complicated, because with the absence of all those other levels of regulation, and being that transcription is so important for regulation, it's really important that these things exist. So really, there are two important components to this. There is the cyclic AMP binding site, which exists upstream of the promoter region, and the promoter region lying downstream of the operator, which can bind a repressor, which can be alleviated by the presence of lactose. So the system really is basically, can we turn on genes that can break down other monosaccharides, or maybe even disaccharides, in the absence of glucose, so that way we might be able to anabolize this particular type of sugar or catabolize this kind of sugar into glucose. So in the absence of cyclic AMP, and just really quickly, cyclic AMP is a byproduct of having a high amount of AMP, and you only have a high amount of AMP if you have low glucose. Because if you have a lot of glucose, you would have a lot of ATP, right? So glucose results in ATP, and AMP is the byproduct of the loss of AMP. So AMP is the loss of ATP. So in the absence of cyclic AMP, the CAP protein can't bind. So if there's no AMP, you cannot make CAMP, or cyclic adenine monophosphate, excuse me. And therefore, what you get is RNA polymerase that can read through, but the transcription occurs at a low rate, meaning that in this case, there is no lactose. So CAP doesn't bind the promoter. but there's also nothing on the operator, right? There's no repressor on the operator, so you get some level of transcription, but it's not synergized by anything. In the case where you have plus lactose and plus CAMP, so this would mean that there's low glucose, right? There's low glucose. So you need to find a way to get glucose if you have low glucose, and you're getting it so you don't have a lot of energy, which is why you're making CAMP. And in the presence of lactose, the operator is no longer bound by the repressor, so that's gone. In CAMP, binding cap protein allows it to bind that cap site, and this enhances transcription, so it increases RNA biosynthesis. In the presence of CAMP, sorry, and also just kind of to follow this up, this also increases RNA polymerase activity. So the same system. Now, if on the other hand there is no allolactose, right? So there's no lactose, and there's a high amount of CAMP, so that means we also have low or no glucose. That means that you get two different things. So CAMP binds to the cap part, which enhances RNA polymerase binding and would normally increase transcription. However, the presence of that repressor causes the RNA polymerase to stall, and then eventually the RNA polymerase complex to dissociate, and therefore there's no transcription. And so RNA synthesis is blocked when this occurs. So I think I'm going to cut it there. Let's see. Sorry. All right, I'm going to cut it there, and we will pick up the rest of this on Monday.